The human brain is one of the most complex pieces of matter known in this universe, and many questions can be asked. How is this complexity so reproducibly programmed in each and every one of us? What are the molecular machinery that coordinate this process to give us features like personality, creativity, and emotions? One of those mechanisms is the ability of our brain to program the production and the organization of our nerve cells in the correct locations in the brain. This all occurs in the uterus in a highly coordinated fashion that we now understand thanks to many genetic and neuropathological studies. The goal of today's episode is to review some of the molecular machinery that drive neural migration from the area where they're created all the way to the ends of the brain, the cortex, where the neurons reside permanently throughout our whole life. In addition to reviewing the normal physiology of this process, we're going to see what happens when there are genetic mutations that perturb this process and lead to a number of diseases known as neuromigrational disorders. These are very complex and devastating disorders that can occur in newborns. So I hope you enjoy this lecture. Let's get to it. My name is Phidias Diamandis. I'm a neuropathologist at the University Health Network and a assistant professor at the University of Toronto. Today we're going to be talking about one of my favorite topics, neural migration, providing an overview of the theory and of course the disorders that arise when things go wrong. I'm supposed to be giving this presentation to some of the residents in our neuropathology program, but I thought I'd also share it here on the channel. So, of course, disclaimer, this is for educational purposes only. It's not a diagnostic tool by any means. I'm an adult neuropathologist with really more of a hobby in neurodevelopmental disorders. So please keep that in mind. So today we're going to go and cover an overview of normal neural development with a focus on some of the molecular players of neural migration, and then kind of use that theory to try to understand the wide array of neural migrational disorders that have been characterized. So let's begin with, again, just an overview of neurodevelopment. This is a little cartoon one of my former master's students, Jennifer Cow, created. And basically, as we know, neurodevelopment occurs almost entirely in utero, and really our brain begins to be formed after the fourth week of in utero life during a process called neurulation, where you have this neuroectoderm, this one of the three germ layers, fold up upon itself to form this neural tube that will go on to form the spinal cord and cerebral cortex and cerebellum, and so on and so forth. So very important structure, and once it's formed, it's at the fourth week stage. It immediately begins to form the brain through progenitor proliferation, differentiation and migration of the neurons to the correct location. So once this unilayer structure is formed, there will go a massive proliferation potential to form these germinal centers or ganglionic eminences that really house a large amount of neural precursors. And while they're being produced, they undergo neurogenesis to form neurons. And shortly after, in a kind of coordinated process, these elements will migrate from this ventricular zone, which is the hollow aspect of the tube, to the most distal parts of the brain to form the cortical plate. And as we'll see, there's two types of migration. There's radial migration from this dorsal part of the subventricular zone, and there's also a tangential migration that occurs from these medial and lateral ganglionic eminences. And really, that occurs in the first 20 weeks of gestational life, and it's a profound statement. But at that point, most of our neurons are not only formed, but they're in their correct location for almost the rest of our lives. So what happens then? The brain continues to grow by changing its developmental program in these germinal zones and producing glia. So glia are astrocytes and oligodendrocytes that are needed to support the circuitry of the neurons. And of course, there's additional maturation steps in the cortex where things undergo neuronal maturation. Synapses are formed. There's dendritic maturation and synaptic pruning, and of course myelination to form the kind of the adult human brain that we're all familiar with. So obviously a very complicated process, and many things can obviously go wrong. And when they do, there's a number of diseases that have been attributed to defects in one of these complex processes. And of course, the one that we're going to talk about today is neural migration, and really the diseases that are hallmarks of neural migrational disorders are Lizencephaly, polymicrogyria, heterotopias, epilepsy, and forecortical dysplasia. These will be saved for a different lecture, just to kind of keep it a reasonable length. So there's some key points that I want to emphasize that are important to understand. Is that at 4 weeks, you have this ventricular zone and a relatively thin preplate, as it's called, which has a very small number of neurons. The preplate neurons and the Cajal-Retziat cells, these are kind of the founder neurons that are produced in the ventricular zone, and the first neurons that kind of exit this this key structure, this progenitor region. As we know, the neurons lie in the cerebral cortex, which is the surface of the brain, and thus it is they're traveling quite some time. So errors that occur can occur at any point during their migrational process. So as I mentioned, you have your preplate and Cajal-Retziat cells. Once these are formed, these proliferative zones will generate what are called radial glial cells. These are your kind of definitive neural stem cells that really extend a process to the glial imitans here at the peel brain surface, and also extend a process to the ependymal zone to form this kind of radioglial scaffold, which provides a nice route for newly formed neurons to migrate this large distance in a kind of very coordinated fashion towards the subplate, splitting the subplate and the marginal zone, which is where your Cajal-Retziat cells are, to populate the six-layered cortex. Now, interestingly, the cortex has a characteristic pattern known as an inside-out pattern of generation, in which these migrating neurons will first populate the inside layer, or layer 6 and 5 of this cortical plate, and then newly formed neurons will reside at a superficial level to these original neurons. And thus you can actually date the neurons by understanding where they are located in the cortical plate as to their age relative to one another. Now, how do they know where to stop? One of the key ways is that these Cajal-Retziat cells secrete a protein called relin, which binds to a receptor known as the very low density lipoprotein receptor on migrating neurons, and tells it that you've reached the top and it's time to jump off, so the next neuron can go. And because of that phenomenon, new neurons really have to go to the edge of the cortical plate to be subjected to this renin secretion to tell them to stop migrating. And that's really why you have this inside-out pattern. And we'll see what happens when that becomes defective. So some key points here to note that will hopefully help make this process more understandable as we go through the notes is that, again, neurons reside away from their site of birth. So they're born in the ventricular zone, and really you can see they have to travel a long distance to reach their final destination. And as the brain is maturing, new neurons have to travel even further than the original neurons due to the brain growing, but also having to travel that extra mile for this inside-out pattern. Now, to do this again, they need a lot of complex migratory machinery. There's extracellular matrix proteins, there's adhesion molecules, cytoskeletal proteins that help maintain the shape of the migrating neuron receptors, and of course secreted molecules as we talked about. Things to note, again, there's both radial and tangential migration. The excitatory neurons really arise from this dorsal subventricular zone and radially migrate to form your glutaminergic neurons or your pyramidal cells that really form these long connections, and then your kind of GABAergic neurons arise in this medial ganglionic eminence and carry out this tangential migratory pattern into the cortex, and then will undergo radial migration to find their correct location. So again, these orange ones are undergoing radial migration, and then these green interneurons or inhibitory neurons are undergoing tangential migration, and you'll see why that's important as we cover some of the many diseases of neural migration. One thing to note is, although it's not clearly figured out yet of how the brain forms gyri, it's understood that some of the theories suggest that it's the connections that neurons form that actually drive gyrification or the gyri that we see in normal brains, and thus if you have abnormal neurons, these types of gyri may not form due to this insufficient environment to foster these connections that lead to cortical folding. So one of the hallmark features, as we'll see, of neuromigrational disorders is in fact also the lack of folding that occurs after neural migration has completed. So it's always important to have a framework to remember these different diverse diseases. So I created this summary slide that hopefully you can ingrain in your brain of how I memorize these or try to think of them in a logical way. And basically, you can think of neural migrational disorders as a kind of spectrum of defects in migration. You can have no migration, so cells do not migrate out of the ventricular zone, and when that happens, we get a disease called periventricular nodular heterotopia. You can have impaired migration, where the neurons will migrate towards the cortical plate, but will do so either in a very slow or uncoordinated fashion, and lead to defects in this inside-out pattern, but also in this nice organized lamination pattern. You can also have partial migration, where some neurons migrate normally, but others do not. And as we'll see, that leads to a disease called subcortical band heterotopia through some interesting mechanisms. So if you can have under migration, well, why can't you have over migration? And that's really what happens in cobblestone type 2 Lizencephaly, as opposed to type 1 in impaired migration. These are different diseases, as we'll see. And then a kind of more subtle version of this is called leptomeningeoglial neuronal heterotopia. And then you can have disrupted migration, where it's still unclear what the exact mechanism is, but perhaps there's an insult that occurs during the process of migration that leads to this disease called polymicrogyria, or focal cortical dysplasia. So let's look at those separately. Let's start with what happens when you have no migration. And really, this is the disease called periventricular nodular heterotopia. So here's an image from this very nice website that I recommend you all use for your studies, neuropathologyweb.org, that really highlights the disease very, very nicely. So clinically, this can occur unilateral or bilateral. So on one side of the brain or both. It can be focal or diffuse, and really patients present with seizures. And really the clinical picture, as you'll see from many of these, is quite wide depending on the severity of the mutations and the defect. It can be an incidental finding. Patients can be actually quite normal. It usually only occurs in females, as we'll see why, and usually when it occurs in males, it's incompatible with life. But there are cases where males can have this pattern, as we'll see, due to very mild mutations or mutations in non-canonical genes involved in this process. So if you've never seen this before, this is a brain scan. And as you'll see, you have your gray matter here at the periphery, and then you have your white matter, which is white. Now, what you may appreciate is that in the normal brain, the ventricular zone is lined by a very thin layer of ependyma that's not very visible on brain scans. But here instead, it looks like it's lined by a gray matter-like substance that looks like has the characteristics of the gray matter. So this is your subcortical. This is your periventricular nodular heterotopia. It's periventricular, as the descriptive name suggests. It's nodular, and it's heterotopia. This is brain tissue not in the correct location. And you can see that, again, no migration. These neurons are exactly where they were born, in the ventricular zone, and didn't migrate out. Now, why did some migrate out and some didn't? We'll see that when we talk about the pathobiology. So this is either a familial or sporadic loss of function mutation in a gene called filament A, which is on the X chromosome. The protein filament A is an actin-binding protein involved in cellular structure and migration. So when it's defective, cells are unable to migrate properly, and affects things like neural migration. And thus you get this failure of migration and this heterotopic differentiation of neurons. So it's X-linked dominant. So males only have one X chromosome, so when they have a filament A mutation, all the cells in their brain are unable to migrate out, and thus they cannot form a cortical ribbon that would be required for a basic function. Interestingly, females, due to a process called lionization, where you have this random X inactivation of one of their two X chromosomes. If this was random, half the neurons would actually have a normal filament A protein, while half of them would not. And as a result, half of them are able to migrate to their correct location, while the other half remain in the ventricular zone and form these nodular structures in the ventricular zone. Usually this would be smooth. And that's why this is a disease found primarily in females, just because the male phenotype is incompatible with life. There are, however, hypomorphs or other mutations, as we'll see, that can foster this type of sufficient migration to make males also compatible with life. So again, the rules keep changing as we see more and more of these cases. There's a lot of, as you can imagine, cytoskeletal structure is an important is important for all cells, and thus many of these disorders can also be seen as part of a constellation of multi-organ syndromes in patients with these mutations. So histologically, we saw some gross images. This is Luxel-Fast blue H&E stain, where the Luxel-Fast blue will stain white matter, and the H&E will primarily stain the white and gray matter. But here you can only see the gray matter because of the Luxel-Fast blue staining of the white matter. And you can see you have your cortical ribbon here, which is what you would expect. You have your gray matter, again what you would expect, but again here in the normal human brain, this is completely a very thin structure, and you have these kind of heterotopic nodules of gray matter. So this is very, very in keeping with periventricular nodular heterotopia. These islands are disorganized. They have neurons. They lie under a kind of ependymal lining, obviously, which is the lining of the ventricles. And because, again, these are forming in a somewhat coordinated fashion, you get these kind of rudimentary laminations where you have different types of neurons being generated with respect to their developmental age. So the overlying cortex can be dysplastic and show polymicrogyria, but it can also be normal in females just because of this process where half of the cells are able to, on average, are able to migrate in a kind of normal fashion. Here's some more pictures from another great book, Developmental Neuropathology, also a book I used during my residency. Again, we have this gray matter here. We have white matter. This is a kind of formalin-fixed brain, it would appear. You have your periventricular nodules here. And you can see that they're very bumpy in the ventricles. Now, one teaching point that you don't want to overcall this by normal brain structures. So keep in mind where things like the basal ganglia should be, just to make sure that you don't get overexcited and call a normal subventricular gray matter structures nodular heterotopias. And you can see here on histology you get these nodules. They're very disorganized, and again they will line the ventricular zone. This is include this. This is a more recent paper in this journal called Development that really highlights some of the kind of current knowledge of different mutations that can cause periventricular nodule heterotopias and other migrational disorders, as we'll see as we cover the other ones. Here's our filament A, the kind of classic gene involved in this disorder, but of course there's many genes that have now been implicated in this disorder. So the literature is always changing. Reassuringly, when you look at the main function of these proteins, which is a common theme here, is that it's not random proteins involved in different functions. It's really the migratory machinery and the cytoskeletal structure that are the major kind of biological pathways implicated in these diseases. So let's talk about kind of a milder form, as you may. Milder in terms of biology. Obviously, these are much more severe diseases from a disease like periventricular nodular heterotopia, where females can actually have very subtle findings. So let's look at some of those disorders. So classic, we'll begin with kind of classical type 1 Lizencephaly. This is a pretty rare disorder. Lizencephaly is Greek for smooth brain. And as you can see here, we have this very smooth brain lacking convolutions, except for this Sylvian fissure. Which is highly abnormal depending on what stage of development you're in. So it's quite rare, one in a million, but has very distinctive genetic and pathological findings. So considered exclusively a genetic disease, as we'll see, some of the diseases that we talk about can have both a genetic and acquired pattern. The kind of reported incidences are equal in males and females, but there are two X-linked forms, as we'll see, that occur or are more severe in males. So, probably because of of how rare these disorders are, um, um, they just the additional burden that may occur in males does not come out in studies. Severe psychomotor and intellectual impairment, intractable seizures, dysmorphic facies, as we'll see, especially with this Miller-Dieker syndrome, where you get bitemporal hollowing, hypertelorism, frontal bossing, many other facial dysmorphisms, and so on and so forth, and even congenital heart defects and defects outside the brain. So, multiple fingers. So this is a very special disorder, and we'll see the kind of genetics behind it. ARX is another form of this disorder that's good to know. It's one of the X-linked disorders, and usually manifests with ambiguous genitalia and small adrenal glands. You can have Lizencephaly with reelin mutations, where you can get what's called congenital lymphedema, where the child is completely full with lymph tissue that's not draining properly, leading to this kind of swollen look. These are really more historic to help you determine what type of mutation and thus what type of genetics the family has to help them with family planning. But now these are perhaps less relevant in an area where you can sequence new cases and understand exactly what mutation exists. Life expectancy is highly variable. Some say 10 years, but really this depends, as all these diseases depend on the severity of the mutation. So more kind of hypomorphic mutations that have less severe phenotypes, patients may have severe intellectual disability, but actually live longer. And of course, if there's very severe mutations, it may be more challenging to the outlook. So what brings all these diseases together? So again, there's impaired migrational machinery resulting in a disorganized, thicker cortex. And really they're defects in radial migration. And I put this asterisk here because one of them, particularly ARX, is actually thought to be a defect in a tangential migration. But for the most part, they're thought to be defects in radial migration. And you can see the list of mutations that are being discovered continue to grow. And they have different types of inheritance patterns, from autosomal dominant, recessive, X-linked, and some de novo mutations. And again, there's some syndromes that are associated with each one of these mutations that help you kind of localize, based on the clinical findings, what the presumed mutation is. Now, what do all these genes have in common? They're all involved in the migratory machinery. So imagine this is the molecular machinery on these migrating neurons, and really the way these cytoskeletal proteins work is by trying to pull, not only extending the leading process, but also pulling the nucleus as the neuron migrates. And to do this in a coordinated fashion, you need to have very strong cytoskeletal proteins like your microtubulins, and then you have to have these motor proteins, these dienes, to really drive the pulling of the nucleus and migration. And when you have defects in proteins involved in this complex, like DCX, which is a kind of a classic one, LIS1, 14EE3 epsilon, which is the protein implicated in Miller-Dieker syndrome, tubulinopathies that have mutations in tubulin, you get Lizencephaly type 1 if it's a severe mutation. Now, interestingly, there's this other machinery, as we talked about, called the VLDLR receptor, which is the very low density lipoprotein, which is really the receptor that binds renin and tells the cell to stop migrating when it reaches the top here at the Cajal-Retziat cells in the marginal zone. And so mutations in this will obviously also lead to migrational defects, because this ability to jump off and move out of the way to allow new neurons to come in is significantly impaired when you don't have a stop signal. These will keep migrating and form disorganized kind of migratory kind of a car crash at the top of the of this radioglial scaffold. Now, interestingly, when you look at some proteins close together, some of the molecular phenotypes, some of the clinical phenotypes are very similar. So when you look at your VDRL, you get cerebellar hypoplasia. Renin, which is the ligand, also has cerebellar hypoplasia. And CDK5, which is just immediately downstream, also has cerebellar hypoplasia. So although this can be seen as one unit, there do seems to be some intricacies that lead to additional things in addition to Lizencephaly when particular regions of this molecular machinery are impaired. So let's look at some of the kind of defining pathology. So again, you have Lizencephaly, or a very smooth cortex that lacks cortical sulci, and usually there is one sulcus, the Sylvian fissure, that remains in some of these brains. Just for comparison, this is a normal brain, and you can see this is what normal gyri look like for those newer viewers. And this is the this is what a smooth brain looks like. So quite dramatic. Now there's different spectrums. So Lizencephaly is reserved when you have no gyri, and in fact is often synonymously used with agyria, whereas the term pachygyria really is reserved when there are fewer gyri. Pachygyria actually means thick gyri, but like I said, it's there's a spectrum where you can have 0 to some, depending on the kind of severity of the mutations. For whatever reason, medial. So this is a good example of what a thick cortex looks like. So this is a normal brain. This is again another great book, Ellison and Love, reference text and atlas, a must for all neuropathology trainees. You see the thicker cortex compared to the normal adult brain with less white matter. So this is typical. Again, not only is it thicker, but you don't get any of these gyral patterns because you really need normal cortex to undergo gyrification. For whatever reason, most medial and ventral gyri seem to be spared. So you can see here there's a hint of a cingulate gyrus here, which is interesting. Definitely something you can look for. So thickened cerebral cortex, 1 or 2 centimeters compared to what's normal, 0.3 to 0.5. These are, I think, MRI kind of measures. And then you have this intervening white matter band, which we'll see nicely on additional images. You have enlarged ventricles because of the decrease in the white matter. And again, you can have these cerebellar hypoplasias that should make you suspicious for renin, CDK5, and VLDRL mutations. So here's another nice picture, really highlighting the thickened cortex. And again, the kind of classic appearance is this thick cortex interrupted by this white matter thin myelin strip. I'll give you my theory of why I think it's there in just a little bit. But then you have this reduced white matter and enlarged ventricles. Here's some more nice pictures to really highlight the point. This seems, again, a smooth cortex. You have a nice Sylvian fissure here. This is a Miller-Dieker syndrome, which, as we'll see, is just a variant of the classic LIS1 autosomal dominant mutations that occur. And then when you cut this brain, the vast majority of the diameter here is of the radius from the ventricle is this thickened cortex with very few convolutions. And again, here perhaps there's some convolutions occurring again medially. Interesting. So microscopy. This is a neurofilament stain, really highlighting the abnormal architecture of this brain. Usually we have a 6-layered cortex, as shown here. You have your molecular layer, which has your Cajal-Retzied cells early in life. These eventually vanish. You have your external granular cell layer, your external pyramidal cell layer, your internal granular cell layer, your internal pyramidal cell layer, and then your polymorphic layer here. So this is the normal cortex. And again, this occurs in an inside-out fashion. So these neurons are younger than these ones, but they probably were brother and sister because they're derived from the same radioglial cell that form their scaffold. And then in Lizencephaly, you have this classic 4-layered structure where you have this preservation of the molecular layer that contains the Cajal-Retzied cells. You have, instead of a granular cell layer, you have your this disorganized pyramidal cell layer. Layer 3 is this paucicellular white matter layer, and then you have this 4th layer that's really broad and made up of partially migrated neurons. Now, so there's some interesting things to I think really appreciate here. My suspicion is, again, this is just a theory or how I kind of try to understand it, is that the majority of white matter that we have is made by these projection neurons in the cortex, or layer 5. And so so because layer 5 is now displaced here, it's disorganized. You have a thin, not fully thick white matter layer just immediately beneath it that are probably axons from these pyramidal cells that are kind of aberrant and trying to at least carry out their normal function. Now, the other thing to note is this is completely inverted, right? So the neurons that are supposed to be newly formed and in the outside position of this inside-out pattern, it's in fact inverted. And why this is the case, in my opinion, is it's because as the brain is growing, the distance to the Cajal-Retzied cells gets further and further away. So actually, the newer neurons are given the task to travel the furthest distance compared to their older counterparts. So obviously, if there's a defect in migration, it's going to more negatively affect the newer neurons because of this extra distance that they must travel, and thus this creates this kind of very broad and inverted cortex. Other things to note is that there's simplification of other structures like inferior olivary nuclei and the hippocampus. This is probably not entirely unexpected, given that these are highly intricate and complexly organized structures. So if you have defects in the migratory machinery, you would expect that such structures would be difficult to form. Again, there's cerebellar dysplasias in some cases, and again, cortical spinal tracts are very going to be difficult to form when you don't have very nicely oriented pyramidal cell layers. Here's some H&E images showing the microscopy again. You have this medially, looks actually quite normal. This is the corpus callosum. This is the cingulate gyrus, and then medially it's quite normal. And then as you go laterally, it gets thicker. You have this abnormal layer 3 band here, and you get these kind of these islands of inappropriately migrated neurons. Here's just another high power view of this from Ellison and Love, really showing the white matter in layer 3. So this is a nice picture that really highlights the different morphologies based on genotype-phenotype correlation. So again, the common theme here is that the more we learn about them, the more genes we implicate, but I think the key ones are really LIS1, which is your autosomal dominant. You have your DCX and your ARX, which is your X-linked, and then you have your tubulin mutations that are also tubulinopathies that cause Lizencephaly. I would add relin into this just because of its unique biology. I think that's important to know. But this, I think, cartoon really highlights some key features. So this is the normal cortex. You have a very tight and thin, normally thin cortex, and you have an abundance of white matter because of these nicely formed pyramidal neurons. And then as you in LIS1, you have a 4-layer cortex. Again, we said it's inverted, it's upside down, it's 4 layers, and then for whatever reason, one of the clinical correlations is that there's a posterior predominance. In DCX, you get anterior predominance and a similarly appearing cortex. And then I don't know if this is actually accurate, but you can maybe get these nodules as we saw in the previous histological image in the white matter. ARX is special in the sense that it has a 3-layered cortex, and really, as we'll see, it's hard to understand it's hard to really, I think, draw these that are disorganized, but the key feature has a very cellular molecular layer, and that's interesting as we'll note, because tangentially migrating neurons may be using this molecular layer to actually get to their correct location. So this may have a nice kind of biological correlate as well, and then for whatever tubulin mutations seem to be very, very disorganized. So let's see what I wrote here just to make sure we didn't leave anything out. So LIS1 again is autosomal dominant form and can give you isolated Lizencephaly sequence. So just Lizencephaly. You can also have the Miller-Dieker syndrome, which is basically a deletion of LIS1, but the deletion is actually much larger and encompasses genes telomeric, so closer to the end of the chromosome 17, and include a number of genes, including 14-3-3 epsilon, which is part of that migratory machinery that we saw, but seems to create a syndrome where multi-organs and structures are affected. And again, this is more severe posteriorly than anteriorly. We'll see why that's important when we talked about XList. So XList is the X-linked version of Lizencephaly, found on chromosome XQ22.3. And really DCX encodes another microtubular associated protein as we saw in that picture. And in males you get Lizencephaly. In females you get this very kind of similar biological mechanism that we saw in periventricular nodular heterotopia, where females have a kind of more subtle finding, as we'll see, due to lionization. And this seems to occur more severely anteriorly, allowing you to, again, make these genotype-phenotype correlations in the previous era where genomic testing may have not been ubiquitous. Relin mutations, again, these are autosomal recessive. They're associated with cerebellar hypoplasia, lymphedema, right from birth, and neuromuscular problems. ARX is the other X-linked disorder. This is unique in a number of ways. It's a transcription factor, probably involved in migration, but needless to say, it's not a kind of cytoskeletal protein. So that's one of the reasons why it's unique. In males, it leads to ambiguous genitalia because of their small size, making them hard to identify at birth. Again, it's one of these features. It's this kind of disorder that's thought to affect tangential migration rather than solely affect radial migration. So that's another reason why it's unique and interesting. So let's just go quickly through these. Again, Lizencephali, Lis1, this is this protein here, interacts with both tubulin and dynein. It encodes this protein called beta subunit of platelet activating factor acetyl hydroxylase, PAF-AH1B1. And again, this really interacts with dynein that's really required to pull the nucleus during migration along these tubulin fibers. And you can see that you get a similar picture with mutations in some of the dynein genes. Again, Miller-Dieker syndrome is just kind of Lis1 plus, where you have loss of contiguous telomeric genes such as 14-3-3 epsilon, and leads to this sequence of dysmorphisms that we'll see. So this is a case of Miller-Dieker syndrome from Ellison and Love. This is the kind of stereotypical face. You have this frontal bossing. You have these bitemporal hollowings. You have facial dysmorphisms, hypertelorism, polydactyly, and so on and so forth. And then at autopsy, you'll see a very smooth brain, preserved sylvian fissure, thick cortex when you cut the brain in half, and then you get this histology that has a very thick cortical plate with some obvious migrational defects as well. These are periventricular nodules as they would appear. Again, with these lissencephalic pathways, you get defects in these very intricate nuclei, such as the inferior olivary nucleus, as shown here, the hippocampus. So look out for that too when you're looking at the histology. So XList is, again, one of these interesting cases where it's another microtubule-associated protein required for neural migration. In males that have one X chromosome, you get Lizencephali, very similar to Lis1 mutations. In females, because half the cells are able to make up for that defect using the other normal X chromosome, you get half cells with a normal DCX, and thus you get kind of a normal histology in the cortex, and then this subcortical band is shown here on the imaging. That's gray matter within juxtaposed between the ventricle and the gray matter. So this is beautiful here on this section as well from Ellison and Love. You get a ventricle. Here's your white matter. Here's your normal appearing cortex. And then you get this second band of a subcortical band of heterotopia. So again, more severe anteriorly, usually kind of associated with females, but as we learn more and more and see more and more cases, it's now being increasingly seen in males when there's a hypomorph mutation, allowing DCX to function somewhat, but not perfectly. So keep that in mind, that these are classic associations that are not by any means definitive. And the histology of in females, at least, is a normal cortex. Clinical phenotype is variable. Anytime you have ectopic, heterotopic gray matter that's electrically active, seizures is always a risk. These are in fact active areas of neural tissue. They respond on PET on imaging that allows you to see what parts of the brain are active. So these are in fact electrophysiologically active areas of the brain, and really it's sometimes hard to predict the clinical severity just based on the imaging. So there's of course other mutations that we talked about, ARX mutations. This is an X-linked Lizencephalo with ambiguous genitalia. It's a transcription factor, but obviously the phenotype would suggest that this transcription factor is involved in neural migration. It's associated with agenesis of the corpus callosum. Again, the kind of the trivia question is that this gene is involved in tangential migration and not just radial migration, as seen in List1 or XList, and thus produces a different cortical pattern. Interestingly, again, because of this random X inactivation, even though we think of this as a male disease, it can occur in females, but in females it doesn't seem to cause Lizencephaly because of this lionization process, and instead they get epilepsy. So again, histologically, you have this three-layer structure where you have a hypercellular layer 1, which is unique, I guess, compared to the other Lizencephalies. You have this second layer, which is composed of these larger pyramidal neurons, and then you have a kind of more heterogeneous layer 3 underlying this. I think this is from Ellison and Love. I apologize for that error. Again, there are a growing number of genes that are being implicated. Again, most of them are involved in neural migration, but just to keep it current, I added this nice reference here. So moving on to overmigration. So as we mentioned, you can have partial migration or impaired migration in type 1 Lizencephaly, subcortical band heterotopia spectrum, but you can have also overmigration where neurons migrate beyond this marginal zone, which is where your Cajal redsheath cells secrete reelin to tell them to stop. And in that case, you get two types of diseases, maybe a spectrum of the same disease. You have your cobblestone type 2 Lizencephaly, and then you have more focal leptomeningeal heterotopias. So cobblestone type 2 Lizencephaly, this is genetically and pathophysiologically distinct from classic type 1 Lizencephaly. Patients have severe psychomotor impairment, they have seizures, but what's very distinct clinically from type 1 is that they also have ocular abnormalities and congenital muscular dystrophy, which is not a feature of type 1. So if you're a clinician, again, in the past, this would kind of help you determine the type of Lizencephaly that you would be most suspicious about. Patients have hydrocephalus, we'll see why. Equal male, female, all of these are autosomal recessive, as we'll see. And really, the life expectancy is highly variable depending on the type of mutation and the severity of the mutation. So what's the issue in type 2 Lizencephaly? We said it's pathophysiologically distinct, and really it's a disorder of defects in glycosylation or O-mannosylation specific proteins. Glycosylation is a very important process. About 1% of our proteins are thought to be involved in glycosylation, and glycosylation of proteins is really thought to enhance protein stability, mediate ligand receptor interactions, and help shuttle proteins for secretion. So when these are defective, you have you can have aberrations in normal biology. There's two types of glycosylation. There's N-linked glycosylation of asparagine, and then there's O-linked glycosylation of serine and threonine. And really, it's the O-linked that's the problem in this disease. And specifically, O-linked is a very kind of very specific modification affecting a select number of proteins, and one of those proteins is this alpha-dystroglycan protein that's important in forming the glial peel limitans, that outer boundary of where the pia meets the edge of the cortex. And so when that happens, you have these defects in your glial peel limitans, which leads to improper alignment of the radioglial cells, and as we'll see, over-migration into the subarachnoid space of the migrating neurons and glia cells. Alpha-dystroglycan is very, very important in muscle biology. It helps keep membrane integrity by connecting muscle cytoskeletons with external ECM proteins such as laminins, and thus when there's defects in this dystrophin or dystroglycan, you get muscular dystrophy. So this is why these two clinical manifestations co-occur in these patients. Really, to do this, as we'll see in the next slide, you need a number of different enzymes to add these different alcohol moieties or sugar moieties to these serine threonine residues. And so when that happens, when you have defects in those proteins, you get defects in alpha-dystroglycan pathology. So this is a nice kind of picture showing what the defect is. So normally you have your ventricular zone, you have your radioglial cells where neurons migrate, and then they reach the top here where they interact with your Cajal-Retziat cells, and then they jump off to allow new neurons to migrate further. In normal development, of course, this leads to this inside-out cortex that we talked about. In type 2 Lizencephaly, you have these breaks in the glial peel limitans. This leads to your Cajal-Retziat cells extending into the subarachnoid space. It leads to these radioglial fibers extending in the subarachnoid space, and thus migration is also as a result of these abnormally placed radioglial kind of scaffolds will also migrate into the subarachnoid space and lead to this over-migration phenomenon. So there's different genes that can affect this glycosylation process, and these are associated with different syndromes. There's about 16 total genes identified. We're just going to go over the classic ones, which is really Walker-Warburg, muscle-eye brain disease, Fukuyama congenital muscular dystrophy, are the real main ones. And you can see these are the enzymes that we saw in the previous cartoon that are required for this O-mannose transferase or O-type glycosylation. The classic diseases are Walker-Warburg syndrome. This is a mutation classically in this protein O-mannose transferase protein or gene, but now it's I think realized that it can occur in other mutations as well. The key thing here is this occurs worldwide, and that's important because the other two have very specific geographic locations. So if you're practicing outside of Finland or Japan and you see these types of diseases, this is the most likely cause that you're going to be experiencing. This is the most severe as well. Patients survive less than 2 years with this disease on average. So muscle-eye brain disease, again, it's due to this mutation in POMGNT1. This is another type of O-mannose transferase, and really the key thing here is that it's found mostly in Finland for whatever reason. So Fukuyama congenital muscular dystrophy is probably the kind of most common and the most interesting of these because of its biology. So this is due to mutations in this protein called fukutin. And interestingly, the kind of common mutation is shown here. This is the normal kind of mRNA sequence with these 10 exons in this untranslated region, and really the defect in these cases is this retrotransposon insertion into the non-coding region that leads to this aberrant splice site that causes defect in the final exon and destabilization of the kind of mRNA protein, leading to degradation. This is the most common type. It's found almost exclusively in Japan. Incidence is 3 in 100,000. Some references we've seen suggest that it's 1 in 90 Japanese carriers are carriers for this specific mutation. And interestingly, 87% of cases in Japan appear to come from one ancestral founder. Given this unique biology, it's unlikely that this has occurred multiple times, and so it's thought that this is actually quite incredible. Just how common this mechanism is in Japan. This is a little bit milder. It occurs. Patients are bedridden about less than 10 years and die after 20 years. Again, bedridden because of the muscular dystrophy, most likely. But again, they have severe psychomotor impairments and seizures due to their Lizencephali. These are other forms of this disease, again, related to proteins that are important O-mannose glycosylation. So again, nice to see this this ability for the mutations to cluster in a specific family of proteins. So again, O-glycosylation is all the proteins that have been found related to this disease really affect this specific pathway. So microscopy, you have small brains, you have this irregular cobblestone pattern, and where you have again this very thick and white leptomeningeal covering, which is your over-migrated neural tissue. Now, one of the key functions of the subarachnoid space is to help divert CSF into the veins to drain out of the brain. When this is clogged up by these neural elements, these ectopic neural elements, obviously this draining system doesn't work, so you get what's called communicating hydrocephalus, where all your ventricular system kind of backs up and is filled with extra CSF. One of the nice mnemonics ways to remember Walker-Warburg syndrome is this hard E syndrome, where you have hydrocephalus, agyria, retinal dysplasias, occipital lobe, and cephaloceles in 5 to 10% of cases, and that's why it's plus or minus E. You can also have vermeil hypoplasia and Dandy-Walker malformations in 20% of cases. Now, interestingly, deep nuclei are preserved, again, because this is not necessarily a defect of neural migration, but rather defects in the boundaries of where neurons will migrate to the cortex. So these deep nuclei, perhaps because they're not reliant on that renin signal to stop in their correct location, actually form nicely, which is very distinctive from type 1. Again, you have this thick gray matter layer, and then again, whenever you have problems with your neurons, you're going to have a very small amount of white matter, and you're not going to have these nice gyri that you see in the normal brain. So you have a very smooth cortex, you have big and large ventricles, and again, a thick, ill-defined gray matter. So microscopy-wise, Again, it's very hard to identify cortical layers. You have disorganized neurons, and really you have these intermingled bundles of white matter that can also be seen in these different sections. The pia is interrupted. If you stain with collagen, you'll see fragments of pia, and again, you'll see these over-migrated neurons. Now, how do you know that these are over-migrated? Well, if you look closely in this picture here, you have this large, thick vessel. This is way too large to be in the cortex. This is a leptomeningeal vessel on the surface of the brain. So anything above this, you can interpret as being over-migrated. The cerebellum, again, is disorganized. Muscle, if you take a muscle biopsy or an autopsy, you have congenital muscular dystrophy with regenerating and degenerating fibers and fibrosis. And again, the eyes show retinal dysplasias and cataracts. Here's some more pictures showing the difference between a normal and type 2 Lizencephaly. Here you have ventricular zone with some precursors still present. You have this very nice tight cortical ribbon here in the normal, whereas you get this very disorganized and thicker cortical structure here in type 2 Lizencephaly. And again, this arrow really is highlighting these large vessels that have no business being in deep within the cortex. Here's just a picture of the cerebellum, again highlighting that this disorganization is found diffusely throughout the brain. Again, because wherever you're going to have pia, you're going to have this problem. Some more pictures from Ellison and Love highlighting the disorganized brain tissue. And again, this here you have these large vessels. These appear to be disorganized migration of neurons before the glial pia limitans, whereas everything up here is in the subarachnoid space. So very, very profound and obvious pathology shown here. So leptomeningeal nodular heterotopia, this is a kind of milder form of this disease where you have just these kind of focal protrusions of glial neural tissue that appear to have migrated beyond the boundary of the molecular layer and the leptomeningeal space. And it's unclear. These are poorly characterized, in my opinion. Can be caused by focal hypoxic or toxic injuries. Their significance is unclear because they're hard to see on MRI. You can't even see them at pathology. You really can only see them under the microscope, and they kind of commonly occur with other pathologies when you do see them, and thus their significance is, in my opinion, unclear. So again, you get focal protrusions of tissue in the leptomeningeal space. There's usually some sort of central vessel or some radial structure showing, suggesting some normal organization. But other than that, I just leave it here as a kind of milder form of disruptions in this glial pia limitans. So to finish things up, we're going to end the discussion of neural migrational disorders by talking what are often referred to as disruptions in migration. And ironically, these are, in my opinion, some of the more poorly understood disorders, at least polymicrogyria, but it's perhaps the most common neural migrational disorder that we see. We're going to save focal cortical dysplasia for another time in epilepsy, and really just end with polymicrogyria. So this usually occurs once migration has finished, or during the kind of final steps of migration, and as we'll see, you get very specific patterns because of that. So what is polymicrogyria? As its name suggests, it's an excessive folding of the cortical ribbon into form miniaturized gyri-like structures fused together, giving the cerebral cortex this Morocco leather type of appearance. Again, it's quite common compared to some of the other disorders that we've discussed today, 1 in 2,000. And clinically, the findings are going to really depend on the extent and the location of this defect. You can have hemiparesis, quadriparesis. If it's diffuse, seizures are common, developmental delay, psychomotor impairments. Again, really depending on the location and the function that area of the brain contributes to. So the risk factors include both genetic and acquired causes. There's a number of regions of the genome that have been implicated in kind of familial disorders, including autosomal dominant, recessive, and X-linked diseases. They're often seen in other types of disorders, such as metabolic disorders, thanatophoric dysplasias with FGF mutations, and other kind of congenital anomaly syndromes. Probably the more common causes that you may encounter are usually due to either intrauterine torch infections, CMV, toxoplasmosis, syphilis, and any kind of intrauterine hypoperfusion. So any kind of hypoxic ischemic insult where there's blood loss that compromises blood flow during the period of neural migration is thought to cause polymicrogyria, including competitive syndromes, conditions where you have twin or triplet pregnancies and blood supply to each fetus may be compromised. So pathophysiologically, again, more commonly, it's the most commonly acquired malformation and occurs during month 3 to 5. Again, that's weeks 12 to 20, really around the time when neuronal migration is at its peak. That's why it's considered a neural migrational disorder. And it's unclear what the mechanism is, but it's possible that the kind of hypoxic insult or the viral insult can cause damage to the deeper cortical layers and the overlying pia, giving it the, as we see, the appearance that we see. What may happen is that the newly formed neurons will pass through these damaged layers, the deeper layers, and become superficially disorganized, and if there's breaks in the pia or breaches in the pia, they can connect by nearby gyri, forming these kind of fusions between gyri, and you can see here how these miniaturized fused gyral patterns form. One of the characteristic features that you can tell that this is occurring is that there are again these leptomeningeal vessels that get entrapped while these gyri are being fused, kind of helping us understand the sequence of events that are occurring to form these miniaturized gyri. Again, we know that hypoxia is a big driver of this process because they're often seen in vascular territories and watershed areas where the blood flow can be easily compromised. They also border porencephalic lesions, which are much more severe hypoxic ischemic injuries that occur in utero. And again, there's some patterns where the first 4 layers appear to really represent the normal 4 layers of the cortex, suggesting that there's kind of post-migrational destruction of the deeper layers. Other theories from CMV suggest that because CMV has a predilection for the ventricular zone where your stem cells are, destroying the stem cells in radioglia could lead to impaired migration and neurogenesis that could lead to this aberrant, disorganized process. Again,
these are less well characterized than some of the other disorders based on the current available literature. Macroscopically, though, you're going to see an irregular bumpy surface. Again, usually in the MCA territory, perhaps there's some microgyri here. This area is suspicious as well. Really composed of mini fused gyri, giving this, again, this Moroccan leather appearance. It can be diffuse or focal, depending on where the insult is, bilateral and unilateral, symmetric or asymmetric. And again, in 2/3, they're in this Parasylvian distribution with the MCA distribution. Here's another picture, just again highlighting the Moroccan leather appearance on external exam. And again, when you cut the brain, you can see these miniaturized gyri forming again, which gives it its very appropriate name. Histologically, when you cut and stain these sections, you get this very, very convoluted cortical pattern, as shown here. There's entrapped leptomeningeal vessels, as we'll see. But again, really the kind of hallmark feature at low power is this fusion of the molecular layer to form these irregular patterns, which is basically nearby gyri being fused together. So that's very, very characteristic. It's almost like a low-power diagnosis. And once you see it, it's very clear what's going on. There are two histological patterns that have been described. There's this two-layer pattern that really is made up of the fused molecular layers with the central leptomeningeal vessel, and then the kind of disorganized cortical layers beneath it. And then there's a four-layered structure where you have your outer molecular layer. You have two neuronal layers separated by this paucicellular band of myelinated fibers. So again, this is what you see. And again, it's a disorder that's common with metabolic disorders, things like thanatophoric dysplasia. So it's often seen concurrent with other findings, and of course, even in acquired conditions like CMV, look for your viral inclusions, look for other areas of hypoxic injury to suggest a kind of more diffuse process. Here's another H&E LFB really showing this complex pattern of molecular layer fusing and forming this kind of advanced, very complex pattern that is very abnormal. And again, if you zoom in, you're going to see these large vessels encased within the this molecular layer that's fused, which is very characteristic. That gives you evidence that there is fusion of these adjacent gyri. So there you have it. We've kind of covered the neural migrational disorders of early neurodevelopment. We didn't cover the first two. This is a nice table that really summarizes, I think, many of these disorders we discussed. When there's processes affecting progenitor cell proliferation and apoptosis, you can get microcephaly, which is obviously hypoproliferation, and macrocephaly, which is abnormally increased proliferation, and this gives you abnormally small and big heads, respectively. Hemimegancephaly is when it occurs only on one side. Focal cortical dysplasias, we'll see later, are these kind of disrupted lamination patterns with these very atypical cells, such as balloon cells that are known to cause epilepsy. The ones that we really focused on that you should kind of ingrain in your head is Lizencephaly type 1. Again, these are really the defects in the migrational machinery involved in neural migration. You have your periventricular heterotopias with the classic filament A mutations, where cells are unable to leave the ventricular zone, And then you have your subcortical band heterotopias due to DCX mutations, where you get that second band of gray matter in the white matter. Cobblestone Lizencephalo we discussed. This is a problem not with migration, but rather a defect in the glial peel limitans, which leads to overmigration into the subarachnoid space. And again, we have polymicrogyria, which is really the excess amount of convolutions due to this fusion of nearby molecular layers with intervening leptomeningeal vessels. Schizencephaly, again, these are more kind of dramatic lesions that can occur with destructive lesions during development, whether it be hypoxic ischemic injury or some sort of torch infection. But we didn't cover those too closely, but perhaps we'll do that in another session. One thing to take away is, again, these were at one point very discrete and well-defined entities. The more and more we understand them and the more and more we study them, we see that they're in fact somewhat heterogeneous and overlapping. I don't want to emphasize that too much, because again, there is unique biology as we saw, but of course these diseases can co-occur, and depending on the severity of the mutations, can lead to very confusing phenotypes. So just be aware that these nice little compartments that we've categorized the diseases is undergoing evolution. So I want to provide the references and acknowledgments for the amazing photos that took many years to accumulate. These are great books that I used during my training, and where the photos have come from. Another great resource here, neuropathologyweb.org, Ellison and Love, neurodevelopmental neuropathology, and the review paper that we discussed. I also want to mention that we want to acknowledge the University of Toronto for providing funding through an Instructional Technology Innovation Fund Award to really help produce these videos and NeuroPsyQ in general. So thank you. And really, we're going to be using these funds to understand the impact of providing these educational resources in a kind of free and scalable manner to the community. So again, your feedback is very much appreciated as we try to understand the value of making these free and available for everyone. So thank you again for your attention, and see you at the next one.
So there you have it. I hope that was informative. Those were an overview of neural migration and the disorders associated with defects in the normal physiology and biology of these complex mechanisms. I hope you learned a lot, and we hope to see you at the next one. And don't forget to share with your friends, like and subscribe so you never miss an episode.